Hello, so my topic today is about RNA polymerase, specifically chain elongation. So I'm going to break up this presentation into four subtopics as shown on the screen. So let's start with chain elongation direction. The direction of RNA chain elongation was established by determining the rate at which the radioactive label from this molecule is incorporated into RNA. Based on this experiment, they found that RNA chain growth happens in the 5' to 3' direction. This is shown here in the picture. So as you can see here, the 3' OH group allows for the elongation of RNA. This conclusion that RNA chain growth happens in the 5' to 3' direction was corroborated by the observation of the antibiotic corticipin. This antibiotic has a hydrogen on the 3' carbon as shown in red on the picture. Since it lacks a 3' OH group, when corticipin is added to the 3' end of an existing and growing RNA strand, it prevents the RNA's chain's further elongation, effectively stopping transcription as a whole and making it an effective medication. Now let's see a short clip Professor Dave explains on YouTube that shows elongation.
One of the strands will serve as the template strand, or antisense strand, meaning it will be used to generate the mRNA. And the other is the non-template strand, or the sense strand. RNA polymerase doesn't need a primer. It simply initiates mRNA synthesis at the start point and then moves downstream along the gene in a process called elongation, synthesizing the mRNA as it goes, reading the antisense strand from 3' to 5' and generating the mRNA from the 5' end, attaching RNA nucleotides to the 3' end as it goes. This is very similar to the way DNA polymerase synthesizes DNA as it moves along the template strand. The main difference here is that RNA is being synthesized, which, as we recall, will be ribose rather than deoxyribose, and it will have uracil instead of thymine. Unlike replication, RNA polymerase zips DNA back up as it goes, keeping only 10 to 20 bases exposed at a time.
So moving along, now that we know that RNA chain growth happens in the 5' to 3' direction, we need to figure out how this process happens. The most plausible method of elongation is that RNA polymerase moves in a straight line while the DNA rotates beneath it. In this case, the DNA would become overwound ahead the advancing transcription bubble, which promotes positive supercoiling, and unwound behind it, which promotes negative supercoiling. The model presumes that the ends of the DNA, as well as the RNA polymerase, are prevented from rotating by attachments within the cell, indicated by the black bars in the picture. This model is supported by the observations of the transcription of plasmids in E. coli and by an experiment done by Kazuhiko Kinoshita through fluorescence microscopy. The in vivo rate of transcription is 20 to 70 nucleotides per second. Once an RNA polymerase molecule has initiated transcription and has moved away from the promoter, another RNA polymerase can follow suit. The synthesis of RNAs that are needed in large quantities, ribosomal RNAs for example, is initiated as often as is sterically possible, about once per second. This could be seen in the picture, which shows an electron micrograph of 3 contiguous ribosomal genes from oocytes of a salamander undergoing transcription. Lastly, we'll talk about intercalating agents. Actinomycin D is a useful antineoplastic/anti-cancer agent produced by Streptomyces antibioticus. So what actinomycin D does is that its phenoxazone ring system, shown in red, is intercalated between the DNA's central G-C base pairs, causing the DNA helix to be severely distorted. This is a picture of what it looks like, and I'm circling how actinomycin D is being inserted in the DNA. So in doing this, actinomycin D strongly inhibits both transcription and DNA replication, presumably by interfering with the passage of RNA and DNA polymerases. And that's all.